Hi everyone, my name is Courtney Dahlke. I'm a biochemistry student, and today I'm going to discuss insulin's role in metabolism and type 1 and type 2 diabetes. First, some background information about insulin. It is a peptide hormone that is produced and released by the beta cells in the islets of Langerhans of the pancreas. It regulates carbohydrate and fat metabolism in the body by causing cells in the liver, skeletal muscle, and fat tissue to absorb glucose from the blood, especially when there's an excess of glucose. It also stops the use of fat as an energy source by inhibiting glucagon release by the liver. It is a control signal to other body systems. An example of this is amino acid uptake by body cells. And lastly, it modifies the activity of numerous enzymes involved in the metabolic processes. Insulin comes in two forms. First, there is an inactive form, which is a hexamer with a zinc ion in the center, and this is the form that insulin is stored in fat cells. Then there's an active form, which is a dimer with alpha and beta chains that are connected by 3 disulfide bonds. This is a good figure and diagram of the stimulating and inhibitory effects of insulin. Insulin stimulates glucose uptake in muscle and adipose tissue, as well as glycolysis, glycogen synthesis, and protein synthesis. It also stimulates the uptake of ions, especially potassium and phosphate ions. Insulin inhibits gluconeogenesis, glucogenolysis, lipolysis, ketogenesis, and proteolysis. This diagram shows the role of insulin in the metabolic energy network. Insulin and glucagon have alternating or counteracting effects in metabolic processes. And insulin is involved in energy storage as well as energy consumption. It helps create an energy supply and regulate that supply. First, I'm going to discuss insulin release. Insulin is released in response to increased blood glucose levels. For instance, after you eat a giant meal at In-N-Out Burger, you have a lot of glucose and a lot of carbohydrates in your body. So, the body will secrete insulin from the beta cells in the islets of Langerhans. It is secreted in 2 phases. First, an initial rapid release of preformed vesicles. Then there's a smaller prolonged release. The initial rapid release is the main process of insulin release. It begins when glucose enters the cell through the glucose transporter 2, or GLUT2, and is rapidly phosphorylated by hexokinase 4, also known as glucokinase. The phosphorylation forms glucose 6-phosphate, which then feeds into glycolysis and the Krebs cycle to increase the level of ATP and reduce AMP. The increase in ATP has a couple of effects, but mainly it inhibits ATP-sensitive potassium channels, which then depolarize the membrane, resulting in the opening of voltage-gated channels that provide localized pulses of calcium to trigger exocytosis of the preformed insulin vesicles. The membrane potential of beta cells is very sensitive to glucose, and the opening of the calcium channels appears as frequent bursts of action potentials. At a glucose concentration just above threshold, each action potential is preceded by a slow depolarization of the membrane, whereas at higher concentrations, it's more of a continuous. This diagram of insulin release shows the glucose entering through the GLUT2 transporter. It is then goes through metabolism or glycolysis in the Krebs cycle to create ATP, and the ATP to ADP ratio increases. The ATP sensitive potassium channels are then closed, and this results in a buildup of potassium or a positive charge inside the cell. Depolarization of the cell membrane opens voltage-gated calcium channels. An increased intracellular calcium concentration then activates phospholipase C. Phospholipase C cleaves PIP2 into IP3 and diacylglycerol. IP3 then binds to endoplasmic reticulum receptor proteins to release calcium from the endoplasmic reticulum and further raise the intracellular calcium concentration. The previously stored insulin secretory vesicles are then released due to the inside negative charge and the outside positive charge. Next, after insulin is released, it can enter the insulin signaling pathway. The binding of insulin results in cross-phosphorylation and activation of the insulin receptor, which is a receptor tyrosine kinase. Phosphorylated sites on the receptor act as binding sites for insulin receptor substrates such as IRS1. The lipid kinase phosphoinositide 3 kinase binds to phosphorylated sites on IRS1 through its regulatory domain and then converts PIP2 into PIP3. Binding to PIP3 activates PIP3 dependent protein kinase, which phosphorylates and activates kinases such as AKT1. Activated AKT1 can then diffuse throughout the cell and continue the signal transduction pathway. Essentially, it is a kinase cascade that involves enzymatic reactions and amplification steps. The result is increased glucose receptor cells on the surface, or GLUT4, which can then stimulate glycolysis. This process is terminated by protein phosphatases. The 3 main protein phosphatases that terminate it are as follows. Protein tyrosine phosphatase remove phosphoryl groups from tyrosine residues on the insulin receptor and the IRS adapter proteins. Lipid phosphatases hydrolyze PIP3 to PIP2, and protein serine phosphatases remove phosphoryl groups from activated protein kinases such as AKT. So here's a deeper picture of the insulin signaling pathway. As you can see, there's many steps involving many substrates, and there's enzymatic reactions, amplification, but the most important thing to notice is that GLUT4 containing vesicle is translocated into the plasma membrane. That means that the plasma membrane can uptake glucose and begin glycolysis. Next, I'm going to discuss diabetes mellitus. Diabetes mellitus is when glucose is overproduced by the liver and underutilized by other organs. About 5% of the human population suffer from it. There's 2 types of this disease. Type 1 diabetes, or the insulin-dependent type, occurs when autoimmune-mediated destruction of insulin-producing beta cells occurs in the pancreas. This results in low levels of circulating insulin or total absence of insulin, and patients who suffer from diabetes type 1 must administer insulin through injections or other means in order to balance their insulin ratio to glucagon and maintain metabolism. Essentially, type 1 diabetes is a fasting state where the glucagon to insulin ratio is higher than normal. Type 2 diabetes, on the other hand, is the insulin-resistant type of diabetes. It occurs when glucose-absorbing cells are resistant to insulin due to genetics or environmental factors. People with type 2 diabetes can have normal or even high levels of insulin in the blood, but due to obesity or age, they don't uptake the insulin properly. Basically, their insulin receptors do not work. Excess fat results in insulin insensitivity, and this is why obesity is a major cause of type 2 diabetes. So the moral of this presentation is that in order for the insulin release and the insulin signaling pathway to work and metabolism to be maintained in a healthy way, you need to monitor your caloric intake, eat healthy, exercise, and if you're already diabetic, you need to manage your insulin levels proactively. If you don't, you will likely get peripheral neuropathy and not be able to feel your feet and get an ulcer, and then your foot will end up like this. So take care of yourselves. Thank you guys for listening to my presentation. Here are some resources I used to complete it, and have a good rest of the term.